be your host for this presentation. Before I introduce our speaker for today, allow me to share a bit about Adjunctive and our products for cell analysis. Adjunctive, as a leader in life sciences, is very focused on enabling our customers to gain the insights they seek. Our rapidly growing cell analysis portfolio enables deeper, more reliable insights across a variety of cell analysis applications, where investigators and drug developers seek to understand dynamic cellular environments and interactions. This is critically important for advancing applied translational research on vaccine development and autoimmune disease, where a more comprehensive understanding of host pathogen effects and mechanisms of immune system function and response is required. Our presentations today will speak specifically to how our solutions are being employed in that context. I'm very excited to have with us today our speaker, Dr. Shannon Russell. Shannon completed her PhD in Dr. Brett Finlay's lab at the University of British Columbia in 2014. Her PhD work focused on examining the role of antibiotic use in early life, its impact on the gastrointestinal microbiome, and how gut microbes affect susceptibility to gut and lung inflammatory disease. In 2015, Shannon moved to South Africa to do a postdoc with Dr. Adrie Steyn in TB research at the Wellcome Trust HHMI-funded Africa Health Research Institute in Durban. Shannon currently works at the British Columbia Center for Disease Control as a research associate, where she is involved in a variety of genomics projects that improve outbreak surveillance in the province, including integrating SARS-CoV-2 genomics into British Columbia's COVID-19 public health response. For the presentation today, we are also joined with an introduction by Dr. Adrie Steyn, who is a leader in infectious disease research. Adrie heads up the lab at the Africa Health Research Institute and also retains a position in research program at the University of Alabama in Birmingham. The Africa Health Research Institute is a multidisciplinary independent research institute based across 2 sites in the province of KwaZulu-Natal, South Africa. The goal of the institute, because of its proximity to HIV and TB, is aimed to support and facilitate discoveries into the susceptibility of transmission and cure of these diseases. But before I hand it over to Adrie, a couple of housekeeping notes. There will be a question and answer period immediately following the talk, and to help us get to as many of your questions as possible, we encourage you to type in your questions during the presentation. We will also meet in our networking lounge at the conclusion of this webinar to further discuss topics presented today with additional experts from the field. We hope you will join our specialists here or visit us in the exhibit hall. And with that, I will hand it over the presentation to Dr. Adrie Steyn.
Thank you very much, Heather, for the introduction. I have one single slide, and in this slide I will describe our future interest and the current ongoing projects in the laboratory. As you
Hi, Adrie. We seem to have lost your audio connection. We'll give Adrie just a moment to hopefully call back in. Let's see. It looks like we are having some technical difficulties that are being resolved at the moment. So we'll give Adrie just a minute here to log back in. Thanks for your patience. So just a reminder that we're having some technical audio issues with Adrie. And so we're not able to hear his introduction to the presentation. What we may do here is actually just move ahead to Shannon's presentation. So Shannon Russell is going to be leading the majority of the talk today. So we'll give just a minute for the slides to advance to that point.
Hi, Heather.
All right, so with that, let's hand it over to Shannon to begin her portion of the presentation.
Okay. Let me just see. I'm seeing a different cover slide here. I'll just move through Adrie's.
Okay, perfect. Thanks, Shannon. Thanks to the audience for your patience. There we go. All right.
Okay. Oh, it's going back. Yeah, unfortunately, we lost Adrie. Must be the South African connection. It's quite a ways away. But thank you, Heather, for the kind introduction. I'm really looking forward to having the opportunity to share some of the research I was working on in South Africa. Just going back here. So can everyone see the cover slide there? Oh, going back. Okay. So in the work I'll present today, I hope to convince you that by applying a metabolic lens to host pathogen interactions and host immunity, you can gain some interesting new perspectives and identify new types of interventions to treat an old pathogen like Mycobacterium tuberculosis. Okay. This keeps going back to slide 7. All right. Well, I wanted to briefly touch on a bit of background of Mycobacterium tuberculosis. So, unlike a pathogen like, an emerging pathogen like COVID-19, Mycobacterium tuberculosis has plagued humans for more than 100 years. And what I think is really fascinating about it is that it's a very old bug, but it still perplexes us today because of some of its unique properties as a pathogen. It's extremely slow growing, and it has an unusually hardy cell wall. This means that it can hang around in the environment for a significant amount of time. It could be months or possibly years, waiting for an opportune host. So, because of these properties, TB still remains the top infectious disease killer in the world. In 2018, there were 10 million new cases and 1.5 million deaths. To put things into perspective, these numbers currently exceed COVID. However, they are disproportionately distributed amongst low-income countries. The most pressing concern is the emergence of drug-resistant strains, MDR and XDR, multi-drug resistant and extensively drug-resistant strains of Mtb. They're becoming increasingly problematic, and they account for the largest number of deaths due to AMR in the world. This is just a snapshot from the Global Tuberculosis Report put out by the WHO in 2020. And it really shows that sub-Saharan African countries have the highest rates of TB per capita, along with parts of Southeast Asia. And TB is the highest in these areas because of the HIV and TB co-epidemic. So for the rest of the talk, I'm going to zoom in to the southern tip of Africa to show you more about tuberculosis in South Africa. So in 2015, I had the pleasure of joining Dr. Steyn's research group at the prestigious Africa Health Research Institute in Durban to address some very important pertinent questions about TB disease development in an area of the world that continues to be deeply impacted by AMR-TB. So I know that Adrie was going to share some examples of some of the really incredible interdisciplinary and cutting-edge work that goes on at AHRI. I did have a slide here, but I took it out because he was going to discuss it a little bit more. But they do some incredible work with different populations in the area, and a lot of really truly translational work. So we'll talk a little bit about that at the end. And so this is the Africa Health Research Institute, and it's located in Durban. So a little bit about TB in South Africa. South Africa has the 4th highest burden of drug-resistant TB in the world. Durban and the province of KwaZulu-Natal has the highest burden of XDR-TB in sub-Saharan Africa. And 60% of TB patients are actually co-infected with HIV, hence the co-epidemic. It's estimated that over half of the population in South Africa is believed to be latently infected with Mtb. So those numbers look like 55.2% of the population, which is huge. And what we see is that there's about a 50% cure rate for TB, including drug-susceptible strains in South Africa. And this should be much higher. But part of it is due to access to care, which remains a key public health challenge. So access to TB diagnostics and TB treatment is particularly difficult in rural communities. One of the few areas of the world where lung resection surgeries remain part of the standard of care to extend lives of patients with severe TB disease. So this is something that Adrie would probably have touched on in his intro. Is he has some incredible relationships with lung pathologists and thoracic surgeons in Durban, where they do lung resections on a daily basis to extend the lives of patients with AMR-TB, and they work together to try to understand a lot of the pathology that happens in TB disease with respect to AMR and drug-susceptible strains. So they would get a lot of these freshly resected lungs that come into the lab to do sort of further research investigations on them. And so to digress a little bit, I was going to go into the science and chat a little bit about the ingenuity of Mtb and its ability to evade the host immune system. So this is one of the reasons why we still struggle to find ways to treat TB, because it's an incredible pathogen. So there's a central dogma which exists, and it's a very simplified model where an individual would breathe in a Mycobacterium tuberculosis bacterium, and this bacteria would then be taken up by a lung-resident macrophage, and a CD4 T cell, an antigen-specific CD4 T cell, would then interact with this macrophage, producing interferon gamma and TNF alpha to actually induce the antimicrobial activity of the macrophage. And theoretically, that macrophage could then kill Mtb and all would be good. But in fact, it's a lot more complex than that. And so this review figure from Nunez-Alves in 2014 illustrates it really nicely, and that was sort of the central dogma, which involved the macrophage and the CD4 T cell. But now this dogma has been revised to include a lot of other different immune cells specifically T cells, but also neutrophils, and a lot of different inflammatory mediators. So there's a lot of different things happening here, and a lot of different cell types that play important roles in TB immunity. So of course, adding to that layer of complexity is that all of this happens in the context of a TB granuloma, which is in itself an architectural wonder. So here is a pathology slide from a patient with pulmonary TB. And this is sort of zoomed in on one of the TB granulomas in the lung. And the cells that are stained brown are CD8 T cells. So you can see those CD8 T cells decorated on the outside of that TB granuloma. And I think looking at this slide was really interesting to me, and this is sort of where the hypothesis was generated when I arrived in the lab, is that, the central dogma that involves just CD4 T cells, there's just no way that, CD4 T cells are the only T cells involved in the TB immune response. Those CD8 T cells are clearly there, decorated on the outside of that granuloma. Why are they there? And are they functioning effectively? Is there something about that granuloma, that the granuloma microenvironment, that is restricting productive T cell responses? Because essentially what's inside the center of that granuloma are the TB bacilli, macrophages, and other necrotic material. So how can those T cells effectively do the killing that they're meant to do so far away from the source of infection? So that, I guess, begs the question, is the granuloma and microenvironment restricting productive T cell responses due to reduced availability of nutrients? So it's all about nutrients, or at least that's what we hypothesized. So, all right, just a little bit about CD8 T cell responses in TB disease. What's interesting is that humans fail to generate robust CD8 T cell memory during MTB infection. TB patients do not develop protective immunity to reinfection. Is this partly due to the lack of a CD8 T cell response? And most existing TB vaccine candidates generate poor CD8 T cell responses. It's just one of the reasons why we have not yet identified an effective TB vaccine. Could be one of the reasons. So does failure to develop lasting Mtb-specific CD8 T cell responses contribute to MTB persistence? And that was the basis of our central hypothesis, which is that MTB maintains persistence during chronic infection by inducing environmental cues that lead to the metabolic and functional deterioration of Mtb-specific CD8 T cell responses in the host. So we have an effector T cell that should be very energetic, metabolically, has increased cytokine production, lots of proliferation and growth, and is capable of clearing a pathogen. But what happens in the context of a TB granuloma? Could that microenvironment within the TB granuloma actually be limiting productive T cell responses, leading to an exhausted T cell, for example, or a quiescent T cell with reduced proliferation and growth, with mitochondrial dysfunction and hence decreased cytokine production? So effectively, that T cell, which should have been metabolically ramped up and ready to fight, and instead it becomes exhausted and ineffective. So we wanted to look at that in the context of a mouse model. And I suppose, if we were able to characterize that shift in metabolic reprogramming, could we then reverse it with some sort of host-directed therapy to rejuvenate the metabolism of that T cell? And that is something, this host-directed therapy is something that, is receiving a lot of attention in other fields, for example, cancer being one of them, other chronic viral infections like hepatitis. So could we apply a similar thought process to TB? So this is the experiment we did. We had, it was a 3-month experiment where we would infect mice with either H37RV, our pathogenic TB strain, or the non-pathogenic vaccine strain, BCG. So we are interested essentially in comparing the lung resident CD8 T cell populations in 2 different infection models. One, pathogenic MTB with non-pathogenic BCG. So what we did was we infected mice using the chronic model, which is only 100 CFUs via the aerosol route. And we had 3 experimental time points, 2 which were considered early time points and 1 considered late. So the early time points were day 21 and day 35 post-infection, and the late time point was at week 12, so about 3 months later. And at each of those 3 time points, we looked at a couple different parameters. We looked at bioenergetics, we looked at the Cell Mito Stress Test and the glycolysis stress test, which I will describe to you a little bit later. We looked at metabolomics. So we did some looking at central carbon metabolism using C13 incorporation, as well as ADP and AMP ratios. And then we also did a lot of flow cytometry to look at glucose uptake, mitochondrial fitness, and cytokine secretion. So the graph you see at the bottom is charting the lung bacilli over time for that model. So from day 21, day 35 to week 12, you can see that, even though we're infecting with 100 CFUs, the MTB pathogenic mycobacteria seem to be much larger in numbers in the lungs of the mice compared to BCG infection. So I guess that leads us to the question, do these infections, knowing that we have a difference in bacterial burden in the lungs, differentially alter the metabolism of the lung CD8 T cells? So is there an effect on T cell metabolism? So the first thing we wanted to look at were actually inhibitory receptor expression, because these markers have been linked to T cell exhaustion. PD1, CTLA4 are 2 inhibitory receptors linked to functional T cell exhaustion, and KLRG1 is a terminal differentiation marker. We also were interested in looking at, and all 3 of these have actually been shown to be upregulated on the surface of T cells from patients with TB. We wanted to look at those in the context of our model and compare the Mtb-infected mice to BCG-infected mice, and then obviously compared to uninfected. So just quickly, the blue bars are CD8 T cell populations from uninfected mice, and these are all lung CD8 T cells. The green are CD8 T cells from the lung of BCG-infected mice, red from Mtb-infected mice, and the orange is actually Mtb-specific CD8 T cells. So we used a TB 10.4 tetramer to look specifically at T cells that we knew were Mtb specific. So that just gives you a comparison to the total CD8 population. So we found that indeed those inhibitory receptors were increased in expression, as well as KLRG1, specifically, or I guess most pronounced in those Mtb-specific T cell populations that we looked at, so the orange bars. Then we looked at glucose uptake. So there had been some literature showing that PD-1, one of the inhibitory receptors, actually is linked to decreased glycolytic capacity in exhausted T cells. So we wanted to know, because we see this increase in inhibitory receptor expression, do we also see a decrease in glucose uptake in these cells? And indeed we do. So you see the bottom left-hand graph, you see Mtb and the Mtb-specific T cells have significantly lower mean fluorescence intensity of 2-NBDG, which indicates that their glucose uptake is significantly lower than CD8 T cells from uninfected or BCG-infected animals. And then the histograms on the right show just that same bar graph, but reflected in histograms. So over time, we see that histogram shifting back towards our control, which is no 2-NBDG, and those Mtb-specific T cells really have a pronounced decrease in glucose uptake. So, knowing that we have this increase in inhibitory expression, inhibitory receptor expression, and this decrease in glucose uptake, we wanted to examine these CD8 T cell populations using extracellular metabolic flux analysis. So I don't know, I wasn't sure how much detail to give here because I imagine there's a lot of experts in the audience, but essentially XF analysis is an incredible technology that can measure two parameters simultaneously: the mitochondrial respiration and glycolytic function in real time. And so below is a diagram of aerobic respiration in the mammalian cell, and just showing that the XF analyzer can in fact measure two things simultaneously: the oxygen consumption rate, or OCR, which is an indicator of oxidative phosphorylation, and extracellular acidification rate, which is a proxy for glycolysis. So if you are an XF pro, you already know all of this, but basically the beauty is that you can create a cell monolayer with any cell type of interest. In my case, it was about 250,000 CD8 T cells per well, purified from the lungs of infected mice. And what you can do is analyze a number of different bioenergetic parameters at once by injecting different metabolism altering compounds into the cell medium using the 4 drug ports shown there in the figure. You have a lot of flexibility in your assay based on the type of compounds that you inject to shift metabolism in different ways. So there were 2 key bioenergetic assays that we used to measure T cell bioenergetics in vivo in these infected mice. And one of them was the Cell Mito Stress Test, which measures mitochondrial respiration and the cell's ability to cope under mitochondrial stress. And to those of you who are new to this technology, Once you create your cell monolayer, you then inject three inhibitor combinations. One being oligomycin, which is an inhibitor of ATP synthase. So if you look at the graph or the figure below, you have the electron transport chain. And so that oligomycin, in fact, blocks ADP synthase, which provides an estimate of mitochondrial-derived ATP production. Then we inject a compound called FCCP, which is a mitochondrial uncoupler. It pushes the cell to its maximum respiratory capacity, or MRC, and it allows protons to flow through the mitochondrial membrane unrestricted. Then we inject rotinoin and antimycin A, which actually effectively shuts down the electron transport chain, revealing any non-mitochondrial oxygen consumption. So we get this really nice CMST profile shown in the graph on the upper left-hand side. So you can measure those different parameters based on your drugs that you've injected. So this gives you some real insight into the mitochondrial health of your cells. So another thing that we use is a bioenergetic phenogram, which is plotting oxygen consumption rate as a function of extracellular acidification rate. It can be used to illustrate the energy phenotype of your cell population of interest. So whether your cell is more glycolytic, more energetic, aerobic, or quiescent, depending on the ratio of your OCR to ECAR. So it gives you a really nice visual of where your cell type fits in relation to others if you're trying to compare different cell populations or the same cell population in different conditions, which is what we were interested in, essentially looking at the different CD8 T cell populations from Mtb or BCG-infected mice. So these are our results, and I'm showing you the time points, day 21, day 35, and week 12. And what you see is that BCG and Mtb have comparable CMST profiles at day 21 post-infection. So overall, they are less energetic than uninfected cells. But at day 35, BCG cells are regaining their respiratory capacity. And at week 12, these CD8 T cells from Mtb-infected mice are, in fact, kind of flatlining. You can see that they're really at the low end of that graph. They have very little basal or resting OCR, and they have very little maximum respiratory capacity when FCCP is added. And then you can actually see, so at that week 12 time point, I have added the TB10.4 tetramer cells. So these are the Mtb-specific T cells, just as a comparison to total CD8 T cells from the infected mice. I was only able to capture that particular cell population at the week 12 time point because you need a ton of cells. And it was only at that chronic stage did I have enough T cells to actually do the analysis. So that's why you don't see that same cell type in the day 21 or the day 35 time point. So underneath the CMST profiles, we have the bioenergetic phenograms displayed underneath that correspond with each of the different time points. And, you know, same trends, you see the same trends. Essentially, at day 21, there's really no difference in the energetic profile of the Mtb infected mice compared to BCG. But then at day 35 and week 12, there's this separation between the different infections and quite a massive gap between the Mtb-infected mice and BCG and uninfected CD8 T cells compared at the week 12 time point. So this suggests that there's really a significant drop in the bioenergetic capacity of these cells from Mtb-infected mice at the chronic stage, so at week 12. And, you know, I guess essentially, what does this mean? We found it really interesting. So next, we wanted to do a T cell activation assay. We wanted to compare what would happen when to those T cells, you know, in their excess plate, when we activate them with anti-CD3, anti-CD28 coated beads to measure the level of T cell activation in response to these activator beads. So how do the bioenergetics compare in their unactivated state compared to the activated state? And you can see on the left-hand side of that graph, the round circles indicate unactivated T cells from each of the groups, uninfected, BCG-infected, or Mtb. And the squares indicate, you know, we've added those activation beads, and now we're measuring the bioenergetics of these cells after activation. So you do see an increase in the energetic state of these cells in every case. However, the parameters are different, or you know, when we compare. So if you look at the bar graph on the right-hand side, I show you specifically OCR and ECAR. And what we found was that the cells from Mtb-infected mice, they seemed to be, their glycolysis seemed fine. So their glycolytic capacity seemed functional. They were able to ramp up glycolysis in response to these activator beads, no problem. But when we looked at OCR, and I'm just going to highlight it here, the OCR barely changed at all compared to our uninfected mice and our BCG-infected mice, which suggests that there could be a defect in mitochondrial metabolism. So those cells, you know, before and after activation really didn't change their oxygen consumption rate at all compared to the other cell types. That was really interesting to us. So we thought, you know, based on what we observed there, you know, is there some defect in the mitochondria? Is there a reason? Is there a drop in the mitochondrial health of these CD8 T cells during chronic Mtb infection? So we did flow cytometry to zoom in on those mitochondria, basically. We found that dysfunctional mitochondria accumulate in those CD8 T cells during chronic infection. And so the way we looked at that was on, let's see, so the graph on the left, the top left, shows you the percentage of depolarized mitochondria in each of those CD8 T cell populations over time. And what we found, the way we measured that was using two mitochondrial dyes, MitoTracker Deep Red and MitoTracker Green, which is actually shown to you in the scatter plot at the bottom. So if we look at those two parameters side by side, we can actually pull out those cells that actually have depolarized mitochondria or sick mitochondria. And those cells are indicated by the triangle there. They have higher mitotracker green, but lower mitotracker deep red. Mitotracker deep red is able to stain for mitochondrial membrane potential, and mitotracker green stains for mitochondrial mass. So we're selecting for cells that have an increased mitochondrial mass and a decreased mitochondrial membrane potential. So in addition to those depolarized mitochondria, another feature of dysfunctional mitochondria is an increase in mitosocs, or in other words, mitochondrial reactive oxygen species, mitochondrial ROS. So we did an assay using this mitosocs dye to show that, in fact, those mitochondria are producing a lot more ROS, which also could be an indicator of mitochondrial dysfunction. So, knowing that we see these trends in the Mtb-infected mice, that it appears that there is this mitochondrial dysfunction, which is then, having an effect on mitochondrial respiration and mitochondrial metabolism, could we then use a drug that targets mitochondrial metabolism to actually, shift these cells or reinvigorate mitochondrial metabolism in these cells? So we're interested in looking at host-directed therapy. And this, so I've just shown you here, this is a really nice review, which has been published by Robert Mahon and Richard Hafner of the Division of AIDS. And they propose, there's a number of different host-directed therapeutic targets that are FDA approved that are being used for, host-directed, therapies that target specific regulatory molecules, many of them involved in metabolism, that are being, explored for other diseases like cancer or other chronic viral infections. And could we potentially repurpose these drugs for Mycobacterium tuberculosis host-directed therapy? They could be used, as an adjunct treatment to existing Mtb therapy, for example, to shorten treatment time. So this is a nice paper sort of going through different types of host-directed therapeutic drugs that, could have an effect on tuberculosis. And so one of the compounds that came to our attention was metformin. And metformin is it's a widely used diabetic drug, and it targets the AMPK-mTOR axis of energy nutrient sensors that control metabolism in mammalian cells. Depending on the concentration, it also targets GAPDH. And recently, it was published. This is actually from 2014 now. That metformin could be a potential adjunct therapy for TB. This paper showed in mice that it was able to synergistically further reduce bacillary burden in mice infected with MTB. So So we wanted to look at metformin in our mouse model. So this is the experiment. We did a similar chronic infection, as I showed you before, with H37RV, our lab strain pathogenic mycobacteria, and we then treated mice from day 7 post-infection all the way to day 35, 500 milligrams per kg daily in an oral format. And we looked at 2 time points, day 21 and day 35. And we looked at sort of similar similar assays as before, so bioenergetics, flow cytometry, and we were also interested in looking at survival as far as CFUs and then lung pathology. So we've got this is one of the results that we noticed was that metformin actually increases the abundance of Mtb-specific CD8 T cells in the lungs. So that proportion of Mtb-specific T cells actually doubles both in the lung and spleen of these mice treated with metformin. That was interesting to us. So all of a sudden they're producing more CD8 T cells. But what about the metabolism of those T cells? Do we actually see metformin impacting the CD8 T cell bioenergetics? And although we didn't see any real changes at day 21, we found some really interesting changes at day 35 post-infection. You will see that in the CMST profile here, those CD8 T cells from mice that were treated with metformin actually have a lot larger. They have increased basal OCR, but then they also have an increased maximum respiratory capacity and spare respiratory capacity. So those cells that you see in purple, we're actually looking at the purple line compared to the red line there. There's a massive increase in the respiratory capacity, the mitochondrial metabolism of these cells. And that shift is also demonstrated in the phenogram shown on your right-hand side, where cells, that were essentially quiescent, they had a more of a quiescent energy phenotype, have become more energetic. And I guess another thing to note is the comparison between the blue and the green dots or lines, depending on which graph you look at. We can also see that metformin affects the metabolism of the CD8 T cells from uninfected mice, but perhaps to a lesser extent, or a smaller magnitude. That was really interesting to us. We could really see that there was a positive effect on mitochondrial metabolism upon metformin treatment in these mice. I guess thinking about the broader implications of the work, improving the metabolic health of CD8 T cells during Mtb infection has the potential to alter downstream inflammatory signaling pathways. It could lead to a reduction in lung damage by limiting excessive airway inflammation. Perhaps it could enhance the survival or fitness of effector T cells required for control of Mtb, but it could also bolster resistance to reinfection by facilitating the generation of long-lived memory cells. We know from the Pierce group that CD8 T cell or CD8 memory cells, they have incredible mitochondrial respiration. They have really high spare respiratory capacity, and they need that in order to survive for a really long time. So, could an intervention like metformin actually make these T cells more robust and capable of long-lived memory? So more importantly, what happens in human pulmonary TB? And I want to just give you a quick snapshot of some of the work that is happening in Durban in Adrie Steyn's lab. And I'm really only going to give you just a snippet of it. There's a ton more, and it's really interesting. But I wanted to talk to you briefly about this report essay study. And At the moment, actually, I need to check with Adrie. There could be many more patients enrolled at this point. I think at the time that I was there, the sample size was 100 patients, and they were looking specifically at adults with drug-susceptible TB. And this study had 3 specific aims. They wanted to look at neutrophil phenotypic markers in peripheral blood, phenotypic drug tolerance in sputum, and measure, well, the third specific aim was measure the bioenergetic health of monocytes and lymphocytes in peripheral blood. And so I think that third specific aim was what Adrie's lab is really focusing on. And as far as I remember, the study was rolled out in 2017. So some of the work that they've done, let me just move slides here. This was a clinical study that took place in multiple medical clinics in Durban. And essentially at the time TB treatment was prescribed, the patient was enrolled and blood was drawn for a time 0 measurement. These patients were then followed up at 2 months and 6 months, and I actually believe that there is a 12-month time point here, which isn't on the slide, with a blood draw, and that peripheral blood was then purified for monocytes and lymphocytes and run on the extracellular flux analysis instrument to measure their bioenergetic capacity in real time. So the goal of this arm of the study was to understand whether measuring the cellular metabolism of circulating monocytes and lymphocytes can be used as a biomarker to predict TB treatment outcomes. And this is just one slide to quickly show what they found, and I think it's really fascinating. Basically, those, and this is just looking at lymphocytes. So at time 0, before TB treatment, those T cells were basically flatlining. They had very little bioenergetic capacity left in them. And as treatment progressed, we could actually see that the spare respiratory capacity of those cells vastly improved. You can see the CMST profile on the left-hand side. It's amazing, the difference at that 6-month mark that was observed in a number of patients. And you can see on the dot plot on the right-hand side, looking at multiple patients over time, there is definitely a trend of improved spare respiratory capacity among these lymphocytes as treatment progresses. So as the outcome improves for these TB patients. And all of this work was done by Dr. Bridget Cumming and the technician in the lab, Kelvin Adicott, and there could be others at this point that I haven't mentioned, and I apologize. There's an incredible amount of work that has gone into this, and they will have a really, I know that they've got a really nice story that they will be showing you, very soon. So that's just a snippet of what they found there, really showing the translation of our animal work and the application of that, looking at, patient populations with TB, and the impact that TB treatment has on improving the bioenergetic capacity of lymphocytes in the blood. So in summary, I will go through this quickly because I believe time is flying by. What have I told you today? Basically that CD8 T cells from Mtb-infected animals have increased expression of markers associated with T cell exhaustion, and they have decreased glucose uptake. Mitochondrial bioenergetics deteriorate in those CD8 T cells as chronic Mtb infection progresses. Dysfunctional mitochondria appear to accumulate in CD8 T cells from Mtb-infected animals. When we compare MTB and BCG infections, this provides a lot of insight into how metabolism may be involved in the development of protective versus pathogenic immunity. Metformin host-directed therapy has the potential to rejuvenate mitochondrial metabolism in T cells from MTB-infected mice. There's much more we could do here to expand on that as far as future experiments, which we can definitely discuss. And then measuring the bioenergetic health of T cells can be profoundly linked to disease progression and the response to TB treatment. So all that leaving you with the thought that, measuring T cell immunometabolism in TB is really valuable, and it really opens up the door to many future host-directed therapy targets. You could be used as a biomarker, or bioenergetics or type of bioenergetic assay could be used to measure something like vaccine efficacy for new TB vaccine trials. That is the end. Just wrap up by saying that this extracellular flux analysis can really be applied to advancing our understanding of host-pathogen interactions. And, I think that it's an incredible application for really expanding our knowledge of infectious diseases. And I would highly recommend it as a tool. So there's many shout outs. There's so many people that helped with this work. We had many late nights in the lab. Something I didn't touch on, but one and two was the actual, the amount of work that it takes to do these type of analyses in the biosafety level 3 environment is huge. And I couldn't have done it without all of these people in the Steyn lab: Dirk Lamprecht, Bridget Cummings, Ijaz Rahman, Terrence Jones, James Mazzarote, Rakea, Kelvin, Vanessa, everybody. It was such a team effort, and as well as the Animal Care Core, Siobhan and Magani. And I have to thank, obviously, Adrie for being my mentor in all of this and providing so many fascinating insights and teaching me all about metabolism, because I was completely new to the field back in 2015. So and there's tons of funding there that also needs to be recognized. The National Research Foundation, the MRC, South Africa, Department of Defense, NIH, HHMI, as well as the Wellcome Trust. So with that, I'm happy to answer any questions that anyone might have. Thank you for your attention.
Oh my goodness. Thank you so much, Shannon, for such a wonderful presentation. This was really interesting. And again, thanks to our audience today for your patience with the technical difficulties that occurred at the beginning. I believe we have Adrie back on the line. So unfortunately, we're not able to get his introduction, but he's definitely been a huge part of this project as well. But great job, Shannon. So we do have just a couple minutes left, and we do have a number of questions. So we will now start the Q&A portion of the webinar. Have time for a question or two. Just to remind you, if you have a question you'd like to ask, please submit it. We'll be able to get just one or two questions before the end of the hour. But however, if we don't make it to your question, we will be able to forward them on to our speaker and the Adjuvant team to follow up with you. So we'll just hop in. And also to remind you that we will have a networking room after this. So we'll be a networking lounge. So we'll be able to meet and follow up with some additional chats in there. But getting to a question or two on the top before we close. So I just wanted to comment, Shannon, you showed some incredibly interesting data on sort of the response of spare respiratory capacity and mitochondrial function to chronic disease progression with respect to like lymphocyte exhaustion. So that's something that I just wanted to call that out because there's been a tremendous amount of interest around that lately and sort of the applications there. Do you, so now getting to the question, do you have any sense of whether bioenergetics or even any other biomarkers could potentially be used to predict vulnerability or susceptibility to TB infection and disease progression? So in this case, you showed it as sort of a marker for disease progression, but the question is really getting more at looking at someone who is not infected and how they may actually respond to infection as a kind of a vulnerability.
That's an excellent question. I would say absolutely. We already know that, going back to TB, I guess specifically, that there are a number of metabolic disorders that actually increase the risk of TB. And so we think that, having these types of metabolic disorders will then obviously affect the metabolism of your immune system and will increase or make you, more vulnerable to TB disease progression. So there is evidence there, certainly epidemiologically, but then also, looking at specifically that paper that I mentioned from 2014 with metformin, they did do a clinical piece where they actually looked at patients who are on TB treatment as well as getting metformin for their diabetes, and they saw that there was an improvement in those individuals over people who were not on that type of metabolic treatment. So we think that there's definitely a relationship there.
So
interesting.
Thanks
so much on that.
What's that?
Oh, I didn't know if Adrie wanted to comment further. I mean, I think
that
was
evidence. That was a great response. Also, we'll have time to follow up in the networking room as well, if you're able to make it. So I'll just close with one kind of a follow-up sort of question on that. Do you have any thoughts on the mechanisms behind the mitochondrial function that we're seeing associated with this TB infection? Do you think of it as any thoughts on if it's like a direct cause or effect or any mechanisms at play there?
That's a very good question. That is something I think we definitely wanted to follow up with after this publication. I think I don't know if Adrie has an opinion on this, but in my mind, I see it as a nutrient deficiency or something that has to do with that microenvironment where, within the TB granuloma. And I think that I don't think that the Mtb specifically, so the bacterium itself, is having a direct effect on the T cells. I think it's literally the environment that it creates within that TB granuloma. There's probably a direct effect of Mtb on the macrophages, and potentially, because Adrie's lab, Bridget specifically, has shown that there's an interaction, or actually there's an effect from Mtb-infected macrophages on the macrophage metabolism. And so it could be an indirect effect of that, metabolic shift in the macrophage metabolism that is actually having an effect then downstream on the T cells that are coming into the site of infection. And I think that's similar to the behavior of a tumor cell and these tumor infiltrating lymphocytes, where the tumor itself creates this highly glycolytic environment that is really nutrient deplete, and those T cells coming into the environment become polarized and, ineffective at what they're meant to be doing because of that nutrient milieu in the surrounding environment. So I think it's really interesting and speaks to, much more work that needs to be done there.
Yeah, I think that's a really great answer on that. Some thoughts about kind of the comparison between the tumor and microenvironment. Really interesting, amazing presentation, great Q&A that we had time for. So Just seeing that we're a couple minutes over, I just want to thank you again, Shannon and Adrie, for sharing your work with us today. Sorry, again, the technical issues. Thanks to the audience for your patience and questions and participation. So now if you would like to continue your questions and discussion with Shannon and Adrie, then please join us in our networking lounge. And you can do so by clicking in the Discuss Presentation Topics and More kind of poster on the right of your screen, or by clicking the Networking Lounge button at the bottom of your screen. So thanks very much, everyone. We'll hope to see you over in that Networking Lounge. Have a great day, everyone. Thanks, Shannon.
Thank
Thanks,
you.
Bye-bye.
Bye.